I would like to invite Dr. Dhaval to please take over and start today's session.
Thank you, Kalyani. Am I audible clearly?
Yes, you are. Thank you.
Yeah. Thanks for a nice introduction. So let's jump into it. So before going into that, I think you might have been all covered, yeah, like you might have read or heard about coronavirus, and there's no doubt about it. Many people could have heard coronavirus. It's all on the internet. It's all in our daily life. We are hearing about COVID-19 virus, SARS, China, Wuhan, quarantine, vaccine, pandemic, etc., etc. So when to go further, I won't waste a lot of time in going into the biology of coronavirus, as you all are aware with that. But coronavirus in an animated look something like this. But if you focus on the small, red, tiny projections, they are actually spike proteins, which are on the surface of coronavirus, and they interact with the human ACE2 receptor, and then this is the way how corona makes an entry to the human cell. But then other than this spike protein, there are a lot of other proteins also which plays a vital role in coronavirus infection to human cells. So when we talk about COVID, when we talk about COVID, I'm just skipping up the introduction of this COVID and all this thing, just directly jumping to the main part, because I just learned that you have all been introduced to this biology of corona previously also. I'm just skipping all this thing. Moving to the major focus on like what is the R&D role in this current pandemic. So there are three areas wherein we can or biotechnology can intervene, or any biologists can intervene. So the first one is a test. So we can focus on to diagnostic tests like RT-PCR and whatever the case may be, or any kind of point of care diagnostic device. The second one is a treat. So we need to identify any or and develop any therapeutics available to treat COVID-19. So these therapeutics or small molecules should be very efficient in targeting coronavirus proteins. Specifically, there are four important proteins in coronavirus SARS-CoV-2, wherein we can have an excellent, or they are excellent drug target. Coming to this point a bit later on, and the third criteria wherein we can help or where there is a role of R&D is a prevent, and in that case we can develop an efficacious vaccine which can be highly efficient to raise proper immune protection to individual. Moving further, when we talk about drug discovery, I think you might have heard the lecture or previous lecture of docking. So I'm not just going deep into this, but the first job is for target identification, and then we have hit identification, lead identification, then we have clinical trials, and then there is a regulatory process. There is a review and approval. So there are different people involved into this. There are different kind of expertise involved into this, right from cell biology to bioinformatics to pharmaceutical background to clinical practices. There are a lot of various people, various kind of methodology involved into this particular process. Now, while this talk is focused on molecular dynamic simulation, so this MD simulation is obviously done after when you do your docking, like you dock a certain compound which you think is very um, a kind of a lead molecule which will go and bind to the protein target. And then we want to see, when we want to see the dynamic information of this, we utilize this MD simulation for the same. Obviously, MD simulation are utilized for many other processes, but right now this talk is focused on MD simulation for developing therapeutics against the coronavirus 2, SARS-CoV-2. So I'm just discussing more about the way how we can use MD simulation to decipher the dynamics between protein and ligand. So there is a crosstalk or an interplay between these two, and this interplay is often mediated by certain kind of interactions of protein per se amino acid with the ligand moiety or ligand, and there are a lot of kind of bond formation bond there are a lot of kind of interaction energies involved into this. So we'll come on to this later on, a bit later on. As I said, when we go for drug discovery, so we have a lot of compounds and it is not possible to get those compound into wet lab or get those compound into clinical trials, so we need to filter it out. There are many techniques. Basically, initial days they were using high throughput chemical assays or enzyme assays to do the same job. Nowadays, we filter it using docking and simulation protocols. And then whatever hits we get after this docking and simulation, we proceed further with wet lab or in vitro assays, and then we go into clinical trials. So definitely there is a involvement of simulation and docking. There's a computational technique into it. There's a huge domain of this. Now, moving into this SARS-CoV-2, as the talk is about, we need to know, we need to identify what are the druggable targets, that is the proteins into it. Okay, so the first one is a spike protein, and there are a lot of structures available into PDB. Perhaps as per the schedule, I think you are going to see the PDB later on, but there are a lot of structures solved for this coronavirus spike protein into PDB. They have been solved majorly by cryo-EM. That is another kind of a revolutionizing technique by which we can see the entire structure of a protein. But then other than that, we have NMR and X-ray by which we can get some target. Now there is a receptor binding domain on the top of this spike protein, which makes an interaction with this ACE2 receptor, which is present onto human cell. So this interaction is very vital for coronavirus infection into human cell, and many therapeutics or many groups are focusing on to developing any kind of a therapeutics which may be a small molecule or which may be a peptide which can just sit over here and interfere this interaction and disturb the interaction, so in the coronavirus is not able to infect the cells anymore. Now, if you see the entire mechanism of COVID-2 entry into human cell, we do have a lot of proteins, but the major one is the spike protein, as I told you initially. Then we have one important category, that is a proteases. So whenever COVID-2 enters a cell, it's a single stranded RNA virus, and with the help of host machinery, it translates into protein. This RNA translates into protein. And then there is a long ORF, which is broken into small functional protein by the help of protease, which is encoded by the coronavirus 2 itself. So these two are also major important target. And then the third one is a RNA dependent RNA polymerase, which helps to multiply the viral genome. And then there is a packaging of this viral genome into new virons, and then ultimately released from the cell. And then they go and infect the another cell. So this is a routine mechanism of SARS-CoV-2. If you see the proteins, which are the druggable target, there are around 15 ORF, and out of this ORF 1A and 2, 1A and 1B are the longest polypeptide, which are broken into functional polypeptide from NSP1 to NSP11 by and NSP12 to NSP16 by two proteases. One is papain-like protease and another one is a MPRO, which is also known famously as chymotrypsin-like or 3C-like proteases. Then there are a lot of other ORFs which are important for four structural protein S, M, and E. And if we see the target, as I told you earlier also, important one are RNA-dependent RNA polymerase, spike protein, papain-like protease, main protease, and then there are other kind of proteins also. If you see this now, on in this ongoing pandemic, there are a lot of modeling efforts made by a different group of people, wherein they have tried to solve different structures, and like for spike protein also we do have many kind of structures available with us, maybe at different resolution, also with different kind of complex, maybe with complex with RBD domain, complex with spike, sorry, complex with ACE2, or maybe proteases complex with many small molecule, and there are a lot of structures available. I just counted in, I think 10 days back, there were more than 1200 structures available in PDB in recent 1 year for of this SARS-CoV-2. uh, as I told you earlier, there are many important proteins, but if we count or if we want to go for a drug designing approach, these four are very important: spike, RDRP, and proteases. If you see the structure, there are a lot of structures available, just I told you earlier also. I'm not focusing into it right now over here, but I'll just keep this one. And we have four important targets over here wherein we can have a major drug discovery efforts. Now, moving further, I'm just skipping this introduction, but just I want to focus that spike is very important for attachment and entry. These two proteases are very important for cleavage and functioning. The M-Pro is also known as main protease or 3C like cysteine protease. So basically it belongs to a cysteine protease family. And then for amplification, we do have RDRP. Moving further, uh, these are the kind of repurposed drug which are right now proposed for this molecular target. And these are ongoing clinical trials. I'm just skipping it. There are a lot of potential candidate targets into pipeline right now. There are a lot of number of patents in right now filed for this targets. Now I will move on to the actual topic. So this was just a very brief overview of COVID-2 and the druggable targets. Now moving further, when we talk about MD simulation, so uh, it's a very interdisciplinary approach which involves physics, chemistry, mathematics, and biology. So all of the four domains are involved in this particular area of research. And when we talk about simulation, we are trying to simulate some kind of biological macromolecule. In our case, it would be protein or DNA, protein in complex with some kind of molecule or whatever the case may be. If we see the historical perspective, there are a lot of theoretical milestones achieved by Newton, Boltzmann, and Schrodinger. And then we talk about the actual simulation part. Initially, there was a Monte Carlo simulation, and then we had MD simulation. So Rahman is one person, and I'm proud to say that he was an Indian. And Rahman is one of the first Indian who introduced MD simulation. He did an MD simulation of an argon gas. And then we have a lot of many important contribution by these people, and they develop a lot of kind of different force field and algorithm and different softwares. And this contribution was mainly focusing towards protein. So there are different kind of softwares and algorithm and force field which we'll discuss eventually after one or two slides. But before that, why we need to do the simulation? So simulation is only means for accurately determining the thermophysical property of a molecular model system. Now when we talk about molecular model system, it includes all the molecules, which in our case it is from biological perspective. It may be protein, DNA, or whatever it may be. So we cannot, when we have some kind of data, when we have some kind of molecules, and we want to look into that which one is best for, which one may be projected as a therapeutic against a protein target, we cannot directly go for an experimental assays right now. We don't have that much time and money and all this thing. So we need to go for some kind of theoretical calculation, that is simulation. So simulation actually helps you to identify a better lead molecule, a better kind of a therapeutic. Possibly you can shortlist or filter the therapeutics available with you, and then you can take that into a actual prediction, wherein you can design a lot of assays and you can say whether that this molecule is effective against a drug target or not. Now coming to the simulation, there are a lot of different packages famous. I have just listed a few famous ones. So we have Charm, we have Ember, Gromax, NMD, and they do have a kind of specific force field available with them. So Charm definitely supports Charm and Ember. Then Ember is specifically for Ember. Gromax is a little bit wider one. It supports Ember, Gromos, and OPLS. And NMD also supports Cham, Ember, and Gromos. Now, what is this force field about? So, when we talk about force field, I think you might be well aware with the kind of bonded interaction and the non-bonded interaction. So, there are different kind of covalent as well as non-covalent interaction. In non-covalent interaction, we do have hydrogen bond, van der Waals, electrostatic, and apart from that, if you talk about covalent interaction, then they are peptide bond, for example, in case of a protein, or a cysteine-cysteine disulfide bond. Now, whenever an atom communicates with another atom, there may be a kind of interaction, there may be a kind of bond formation between them. So atom can have a different kind of effect on a neighboring atom. So this information, these forces are available in the force field. I have very superficially defined force field, but it is a lot more into that. You can perhaps read that in later on. So when we talk about simulations, I'll just briefly tell you the overview of that. So we do have a topological properties, that is whatever molecule we want to simulate, whether it is a protein that is made up of amino acids. So we do have a kind of a dihedral and torsion angles into it. We do have a topology of all this protein, that is a structure, definite structure of this protein. And the starting conformation of this structure is often provided by X-ray structure or NMR or cryo-EM, or perhaps if you don't have any X-ray structure, then you can also go for a homology model. And then the third one is the energetical property. So that is actual force field, which describes the force acting on each atom of the molecule. So whenever, let's say, for example, this is a molecule. So this carbon definitely have a acting force by the neighboring atoms, that is oxygen in this case and a methyl in this case. So they may have a certain kind of energies involved into it. And when we talk about force field, we do have a torsion angle, we do have a bond stretching force, we do have a harmonic force, and there is a lot of more things into it. And then we talk about the fourth one important property, there is a thermodynamical property. So a sampling algorithm is required that generates a thermodynamical ensemble that matches the exact experimental condition, though we cannot really duplicate the exact real life condition into the simulation, but we are or the algorithms are almost able to do that, but not 100%. But we need to simulate in an environment which is a real life like kind of environment, a proper kind of a pressure, volume, temperature, also needs to be introduced. So these are the four important components of which are, or which we can say these are the four kind of input required for any simulation. So if we see the step, the first one is the initial coordinates which we require from X-ray NMR. Then we do have a structure minimization. That is, we release the strain and we give a time, enough time to relax the molecule, and then we need to do a solvation. Now, why we need to do a solvation is because protein in our body or any kind of biological macromolecule is surrounded by a solvent. It may be inside the cell, it may be on the cell, or it may be on the surface of the cell, or it may be outside the cell. But everywhere around it, there is a solvent molecule into it. And in our body, obviously, we do have water as a major solvent. So we do simulation in presence of water, which is also called as an explicit solvent. But in this case, we can also choose different kind of buffer if we want to do a specific kind of assignment or a job. And then we give some kind of initial velocity. So when the simulation actually starts, there is a kind of a dynamics and vibration involved into it, because those are atoms which will communicate with surrounding atoms. It may be a solvent molecule, it may be another amino acid, another atom of any other amino acid, it may be a communication with the ligand or whatever the case may be. Now, when we talk about proteins, proteins are generally made up of amino acid, and these amino acids are joined by peptide bond, and they are not static as it is seen in an image. So whenever we see an image of a protein, we look or we see secondary structure element like alpha helix or beta sheets or loops, but actually they are not like that. They do have a secondary structure element, but they are dynamic in nature. They're constantly vibrating in nature. So this is what it gives the name molecular dynamics simulation. So what we are trying to do is we are simulating, or we are creating a theoretical environment which is very near to the real life condition, and we are throwing this environment into a molecule which may be protein in our case, and then we are trying to study the dynamics involved into it. And then we heat up the dynamics to a certain temperature, whatever the required temperature it is. You can change this temperature. Then we go for equilibration dynamics. If everything is okay, we go for the final production dynamics, which involve three different kind of ensemble: NVE, NVT, and NPT. And then after you finish your simulation, you get a trajectory. So this trajectory is basically a file or a data which contains the information about the molecules and atom, and also the trajectory. That is, trajectory in a layman word is a motion or a path of any molecule from one place to another, and we do also do get a lot of energy associated with that. So we have a trajectory file with us which have all this information, and then we need to analyze the trajectory, and then whatever our aim is, we can, there are a lot of analysis command, and we can do a lot of kind of different analysis. Now, there are a lot of applications. I'm not just going into that right now in very detail, but the major application or the talk which is focusing right now is on developing a therapeutics against COVID-2 using a kind of a simulation or a workshop in simulation. So what we do in this case is the simulation can actually help us to identify a small molecule, how it is going to interact with the protein, what are the dynamics involved into it, or other way you can also go for simulation of protein into, like, you can study unfolding folding. You can also do a de novo folding process wherein we don't know the fold of a protein. You can just go for simulation and it will try to attain a best kind of fold. There are a lot of applications into it. Now this is the main thing which I need all of you to focus on. So this is the entire layout of a simulation flow chart, we can say. This is specifically for GROMACS. I have adapted from a literature available with us, but this is a very simplified version of a flow chart of a GROMACS simulation. So right now I've been focusing on a GROMACS tool, and I will just show you, demonstrate how to use that. But before going into that, we need to look the entire flowchart of this GROMACS software. So first we start with some kind of coordinates, and that is available from PDB, that is a protein data bank. So NMR X-ray structures are deposited into that. Now we can have a two kind of approach over here. Either you have apoprotein, which is a protein without any other component, without any other cofactor, or without any kind of a ligand, or we do have a protein ligand complex. So whatever it may be. Now, if your aim is doing a simulation after docking, then you will have this protein ligand complex. But you also need a protein as a control, or to compare with it, like how the ligand interaction stabilizes the protein ligand complex. In that case, you also need to do the simulation of an apoprotein. Now, in this case, for example, if we do have apoprotein, like many people are only interested in the folding dynamics or folding pattern or how the molecule behaves, so they go for only apo simulation. And if you really want to investigate the protein ligand dynamics, then you go for a protein ligand complex. Now, let's say we consider that the first case is a apoprotein. We don't have any kind of ligand. So the first step is to strip off any kind of other molecules. Maybe it would be water or any kind of cofactor or any kind of metal ion. Just strip it out, just remove all those things and just subject that particular naked protein into a program called as PDB2GMX. Now let me introduce. GROMAX is a umbrella software or umbrella algorithm wherein you have different kind of programs into it, sub programs into it, and they are utilized at different level of your entire flowchart of MD simulation. So this is the first component is of preparation. So this is called as a first phase wherein you prepare your molecule. So you do have a protein. Now this protein is devoid of any kind of other molecule other than proteins. Then we subject it to a program called as PDB to GMX. So the function of this PDB to GMX is to convert the PDB file into a GROMAX readable format. So it will just take the file, it will convert it into a GROMAX readable format. It will apply all the kind of force field. It will apply what more water molecule or what water model or what kind of solvent atmosphere you want to use, as well as it will generate a topological file. And then we subject it to a configuration program called as an edit conf, wherein we give a configuration like if you want to simulate a protein. Now let's say this is a protein, you want to surround it by a water molecule. You cannot just put a water molecule like this. You need to first create a kind of a 3D box and then add a solvent molecule into it. So this configuration of box is defined by this program edit conf. So it will look something like this. So when you prepare this, it will look something like this. So this is a molecule which you want to simulate. This purple one tiny dots are water molecule or solvent molecule. This is a hypothetical boundary of a box. So wherein you keep the molecule in this box and you keep the solvent and then you try to simulate it. After that, you go for another command which is called as a gen box, which will generate a box and it will add a solvent into it. So this phase is called as a solvation, and this PDB to GMX is called as a force field conversion, wherein we convert the PDB into the GMX. Um, Now, after this, when you have generated a box, you have entered a required amount of solvent molecule into it. We need to add some kind of ions. Now, we all know that proteins are made up of amino acid. Amino acid do have a side chain. All of these amino acid have a different kind of charge depending on the pH of the environment. So over here, we need to neutralize the environment. We need to add the required charge just to make the entire charge of a system zero. So for example, if the protein in the solvent has a charge of +2, then we need to add -2 charge molecule, that is ions, into it. And thus now the entire net system will be zero. The entire charge of the model system will be zero. Preferably in case of GROMAX or in any other tool, we use NaCl to neutralize this. Na is obviously +2, that is Na is a positively charged ion and Cl is a negatively charged ion. So depending on your initial charge, you add the counterpart into it just to neutralize the system. So now till here there is a neutralization. So all of these files and all of these processes are done, and after that we actually jump into an important component called as a energy minimization. So for this there is a command called as an MD run. Now for all these three, for first one is energy minimization, and then the second one is the equilibration, there is a temperature and pressure equilibration, and the third one is a production run. So all of these three, we do have two common command with us, which one is gromp and MD run. So gromp is a gromax preprocessor command wherein we load the system with the kind of a parameters which we want actually. So we load these parameters into it and then we prepare the files and then we go for a final run by command called as an MD run. So there are three important parameter files used over here. The first one is a energy minimization .mdp. So it stands for EM stands for energy minimization. MDP stands for molecular dynamics parameter file. So this is a file which we need to input it. Obviously, we don't need to worry about it. We have a default file parameter with us. But if you want to change some setting, you can obviously do that. You need to read a readme manual of that. So what are the different options available with us? And then after minimization, we do have a equilibration part over here, wherein we have nvt.mdp, that is volume temperature parameter file, and then we have a pressure temperature parameter file. And after that, when the equilibration is done, our system is prepared, our system is stabilized at a certain temperature, at a certain density, at a certain volume and pressure. Then we go for the final md run command. So this is the final md run command parameter file. Again, the same set of two commands, grom, that is a gromax preprocessor, and then final md run. And after the simulation run is completed, we take the trajectory, we process it, and we go for analysis. Okay. So if you want to really break up this entire chart, the first one is a preparation, the second one is a forceful conversion, then solvation, neutralization, energy minimization, and then equilibration, and then the final run. Now this whole scenario is applicable for both apo and protein ligand complex. So if you have a protein ligand complex, there is a ligand present into your system, you just separate it. You can use any, you can do it manually also, or you can write a script for the same. A Python or a Perl script is enough to do the job. So what we do in this case is we separate out the ligand molecule. So now, for example, imagine there is a protein and there is a ligand over here. So what we need to really do is separate the protein and separate the ligand. Protein is subjected to this part wherein we use PDB to GMX. Now about the force field, we do have a forceful information about certain biological macromolecules which are pre-calculated. For example, in case of a protein and DNA, for RNA also. But for ligand, we don't have any force field available with us. We need to generate it by scratch, or we need to generate it using any external software. So in this case, it is a ASPICE, but we do have a lot of softwares available with us, and then you can use those and you can generate the forceful parameter file, and then you can combine this protein forceful parameter, that is a topological file, and the topological file of this ligand, and combine it using, or you can do it manually, or you can use any kind of tool, and then you can again proceed with this exact steps which are common to apo as well as protein ligand complex. So this is all about ligand. So do remember, for ligand, we don't have any inbuilt forceful parameter. We need to generate the topological file externally using any software. Like in this case, it is ASPICE over here. So I just wanted to know that, do you have any kind of doubt in this entire flowchart? Any one of you, any one of the participants, do you have any kind of doubt in this? If not, then we can start with the actual simulation, or we can have a demo. It's very difficult to complete the entire demo in this current session, but I will try my best to introduce with you the software and every other thing. But do you have any kind of doubt in this entire flowchart? You can either write on the comment or you can just speak out over here. Okay. Kalyani, am I audible clearly?
Yes, you are.
Yeah, okay. So the blue ones obviously are the Gromex tools or scripts available with us. In this case, there are user parameters which we can or we need to use it. And the blue purple one are obviously the parameter file. So these are the three important components needed for any simulation program. Now, I'll just tell you that particularly myself, I have used the simulation for many different studies, for many different problems to address some kind of biological investigation. And we have published in very good journals. For Corona 2, there is SARS-CoV-2, we do have 3 publication and 2 more in pipeline. So wherein we have used this simulation technique to just showcase how the protein behaves in presence of any small molecule or in presence of any other component, whatever the interest of area is in our case. So I just move on to the tutorial part. So now first tell me, like before I proceed, I just wanted to check again that, do you have any kind of doubt in the presentation which I made? Uh, Karyani, there is some kind
So
of,
we so we take the questions in the chat box once the presentation is over.
okay,
Then
sure, sure, sure,
we
sure,
can.
sure, sure, sure.
Seems like people are commenting that they don't have any doubts, and there is one question from one of the participants. The question is, can we use homology modeled structure and ligand docked structure for MD simulation, I'm presuming?
Yeah, we can definitely use it, but it's better to use a 3D structure available with us. That is an experimental structure. But if no experimental structure is available, definitely we can go for homology model. We can model the protein. We can then apply a kind of model evaluation tools like Ramachandran plot or many any other tools. We can even minimize the model. We can check whether it is the quality of model is good or not, and then we can choose the ligand, and then we can go for the simulation. There's no second thought about it that we can't use a homology model into this. So is the font visible to you, Kalyani? It's clearly
Yes, it
visible to
is.
you.
Yes, it is.
Okay. So perhaps I hope that it would be clearly visible to many other candidates also. So now I'll just open up my folder wherein I have all these files with me. Okay. So just make it a little bit bigger. Okay. So now right now don't focus on all these files. Just focus on the protein. So what we really do is when I will just open up a PDF and I will show you how to download a PDF file and how to trim all the water molecules and how to do that. I will just show you the different ways for doing the same. You just need to type mpro and what are the protein maybe, but in this case we'll use the mpro of SARS-CoV-2 as an example. So we'll just select filter it out. I will just select mpro for SARS-CoV-2. So there are a lot of structures available with us. I'll just download any one of them. Okay, so now it has been downloaded. I will just copy the file to the desired location. Now if you want to see how it looks like, then we can just open it. Okay, so you can see a pretty uh, animated and a graphical protein molecule over here, wherein you have alpha helix sheets and loops, all the kind of secondary structure in this protein. And obviously, if you see this tiny red molecule, they are water molecules over here. So when you do or when you solve any protein structure using X-ray or any other method, obviously it is into the building crystals or into some kind of a buffer. Probably it would be water in most of the cases. So this water will definitely go and bind onto the protein site, perhaps many other places, maybe in active site or in the surrounding. So they may also get crystallized into it. And over here, which you see in this ball and stick model, this is a ligand molecule. Just select it so that it's visible to you. So you can see over here, I'll just color it in a different way. Okay, so you can see this small molecule over here. It may be perhaps a lead molecule or a therapeutic molecule. Now in this case, there is a there is a covalent bond formation over here. We're not focusing into it, but it depends on the biology of a protein. Now, there are multiple ways to remove any non-protein molecule present in a protein. So you can just select it over here. You can use any tool. In this case, I'm using Chimera. I can just delete it and then save the rest of the file. But if you don't want to do that. So what we can do is just open up the file in any kind of text editor. I preferably use Notepad++, but you can use any one of them. So just go down. You will see I'm not taking the luxury to explain the PDB file format over here, but just I will explain you one line. So over here you can see this is atom information. This is the number of atom, the actual number. This is the kind of atom, that is a C alpha in this case. It is of valine residue. It is from chain A. This is the number of the amino acid. So it is a 296th amino acid, which is valine in our case. And these are the three XYZ coordinates. So it's a Cartesian coordinate system. So wherein you have three values. And when you open up this into any visualization tool, you will see the dot or the location of that atom into that visualization. And then we have a lot of other things. We have not going into this discussion. Now, if you go down, if you scroll at a point, you will find that there is a termination line over here. So it says that all of the amino acids minister, that is 305 in this case. The information about that is ended over here. And now what we do have is a heteroatom information. So this all are molecules other than the protein. So we have DMS, that is dimethyl sulfoxide, DMSO in this case, which would be used in crystallization. And then we have a ligand which is named by R8H, and then we have a water molecule with us. So simply what you can do is just select this entire heteroatom part, go down. There are shortcuts too for the same. We can press shift, control and end, or a down arrow. So we'll do the same. Just go down till this part, press delete, and now you have deleted all the water molecule. Just save this file. So now in our case, if you open up the same file, it would be devoid of any water and the ligand molecule. So I'll just save into a name called as a protein for my simplicity, and just delete this one. You can just label into a name called as a protein. So now we'll go with the first step, that is convert the protein into the Gromex readable format. So I opened up Ubuntu, which I've installed over Windows over here. So this is a command line, and we don't have a time and luxury to go into the Linux right now, but Gromex is often like Gromex executables are available for Linux. There are certain tools by which you can use Gromex into Windows, but they are not that efficient, and they may end up with some kind of you may end up with some kind of an error over here. So right now I am into the path where all my files are available over here. So just navigate into the exact path, and if you do ls, which is again a Linux command, so you can see this much many number of files. Right now just forget about all this file. We'll just focus on to a file called as a protein.pdb. Okay, so the first step is that, as I told you earlier, we need to convert into protein file, that is pdb file, into the gromex file. So we'll just first shoot up the command. Now before going into that, let me tell you one very important website which you can use for learning Gromex from scratch. And this is a very excellent tutorial described with different kind of scenarios, wherein you can use that, and exact if you follow the step-by-step procedure of the same, you will be able to successfully run the simulation. So in our case, we are going to simulate only protein in water. So what we do have is we can refer to the tutorial called as lysozyme in water. Just click over here. There's a lot of description over here. You can just read later on. I'm ignoring all of this thing. And this, you can also utilize the grab command, again do the same job to trip or to trim the protein and to delete all the water molecule or any other kind of water molecule. Now if you go into detail, this is the first command. So what I'm doing is I'm just copying into it and saving into file, and I will later on upload this file. I'll just later on upload this file for your easy convenience. So over here it says that GMX is the name of a program. In our case, it's a Gromax. It's a short form for GMX. And then this is the actual script which we are going to use in the first place, that is PDB to GMX, wherein we will convert the protein file into the Gromax readable file. Minus F is for input. So we are inputting this file. In our case, it would be protein.pdb, which we have saved earlier. And then there is a minus O, which is output. So we'll just replace this into protein.grow. And what kind of water molecule model you want to use. So we have various available water molecule with us. You can use SPC, or you can use tip3, or you can use any other kind of water molecule. We have tip4 also. So in this case, we'll be using it water molecule, water model, which is SPCE. Now, if you run this command, it will instruct that we need to open Gromax. We need to fire this command. We need to input the file, that is protein.pdb, and then there is the output of the same. Now, one, uh, I cannot say a bad thing, but one kind of a problem over here is that Gromax is not a GUI based software. It's entirely operated by command line. So you need to learn this Linux, you need to learn this commands, and by the only way you can you don't have a beautiful windows into this, and so it would be difficult to learn. But if you are not comfortable with the Linux, you can obviously go for Schrodinger Desmond software, which is also used for simulation. I'll just copy this command and I'll just paste it over here. Sorry. Okay, so just pasted the command over here. And before that, I just forgot to mention you that we need to install Gromex into our system. So there are a lot of steps into it, but it's a pretty much easy installation. You can just go into manual and you can install it by yourself also. So when we fire this command, it will ask us, there are a lot of information displayed over here. We need to just ignore those information until unless there is some error. Now it says that what kind of force field you need to select for this particular molecule. So there are a lot of different force field available with us. Few of them are from Amber. We do have Charm. We do have Gromos, and we do have a OPLS force field available with us. All of them now. There is a very a routine question asked by many people that which force field is good, which is bad, or which is not suitable in your case. So you can read this paper. All of these force field have a different advantage and disadvantage, or they are applied in a different scenario, whatever your problem may be. But generally, I prefer to use Ember, or you as an individual can also prefer to use OPLS. It depends. There's an excellent benchmark paper available in PubMed, wherein they have applied different kind of force field into different kind of model proteins, and they have evaluated each one, that which one may be best for which scenario. In any case, if you don't know anything, just go with the 15th number, that is OPLS force field, or you can also go with the ninth number one, that is Gromos 9643A1, or you can go with the fifth one, that is Ember 99SB. Now, do remember one thing. Whenever you are using a protein, for example, and your problem is, let's say you want to see the dynamics of protein at different temperature, let's say 37, 45 degrees Celsius, 50 degrees Celsius, 60, 70, and so on. Use the same force field for each case just to have a better comparison in between them. If you're trying to look whether your ligand stabilizes your protein or not, so you have a apoprotein run just as a control, and you have a protein ligand complex run. So in both of the case, the force field should be common to both of them. The water model should be common to both of them. We cannot use different force field for the same experiment because it will give different kind of result. Okay, so in this case, let's say for example, I'm going with the fifth number, that is Amber 99SB. So I need to just press 5 over here, numeric 5, and just press enter over here. Okay, so it will do a lot of kind of pre-processing, and it will give us a output. Now in this case, it says that you have successfully generated topology from protein.pdb. So now the kind of files, I think I will just copy this file into. Okay, so I'm just copying into a different folder. Okay, so initially we had a protein file, we have converted into protein.grow, and then we have a topologic file generated over here. So now if we open up this file, we'll open up this one, and we'll open up this one. Oh, sorry. Okay, so let's say we had an initial protein.pdb file, which I explained to you. Now we have a GROMAX compatible format. So what the GROMAX or the GROW file includes is the number of atom over here, the residue, the number of that, sorry, the amino acid, the number of that amino acid, the first one that is serine, the second one is glycine, the atom of those amino acid, the number of this, that is 1 to 13 in this case, and then we have xyz coordinate system, and at the end of the file we do have certain values over here. Just ignore it right now. And then we have associated topology file generated from this first command. So it says that it includes a forceful parameter which is available into this file, that is number 99, which we have used. There is only chain A available in this. And then this is the topological information about each and every atom in all of this amino acid. So this is serine for the first one. We have atom type. So in this case, it is N3. So the atom actually is N, that is nitrogen. We have hydrogen, 3 hydrogen. So they have been named H1, H2, H3. This is a number 1 to 13. And then this is charge of that individual atom. So for nitrogen, in this case, the charge is 0.18. We do have a mass over here, and then we have a different time. It's difficult to mention over here. q dot over here in this case, but just assume that it is a total charge. So over here it is 0.1849. Then for the second one, you need to sum up this up. If you sum this up, it will be 0.3747. Then you need to sum this three, it will be 0.56 and so on. At the end of this, somewhere over here, sorry, okay, at the end of this, you will have the final charge of a system. So over here, if you include, if you calculate, or if you sum all the charge of all the atoms present on all the 305 amino acid, it is -4. Okay, this is the bond information. So we're not going into this file format right now. Just keeping this information, then there would be a bond angle information and many other information. If you go at the end of the file, it says that include data topology. So we have used SPC model. Now all this topological information and the file or the required model information are stored into a file which we are just right now importing over here. So whenever the file is read, when it encounter this line, it will go and open this file at this particular location. It will read the required information into it. And then we have this ion file. If you're adding any kind of ion to neutralize the system, or if any ion is already present in the system, but in this case we have removed all the non-protein molecule, but if it is there, if we need to include any ion for our study, then those information are in ion.itp. Now if I'm closing right now this, now we'll move on to the second command. So now moving on to the topology, it's just an explanation what I have explained to you right now. You can read this later on. Now moving into the solvation part. So this is the second command. So I'm just copying it, pasting over here. Oh, sorry. Okay. I'm just pasting over here. Okay. So this is the second command. So I'm just replacing this name with my file names. Okay. Okay. Now do remember the Gromex is whenever you want to run a simulation, as I explained to you, there are a number of steps. So the output of the first or the previous step is the input of the next step. Okay, so output of this, that is protein grow, is the input of this particular command. So here right now again the command which we are going to use edit conf, which is used for generating a configuration. So this is the second phase wherein we are right now. And the input file by minus, we can define it by -f, that is protein.grow, which was an output of the first command. And then in this case, for this command, you need to generate an output file called as protein underscore box. -c is for placing the protein molecule in the center of the box. -d is for defining how far it should be from the edge of the box. So in this case, it is 1 nanometer. So it simply says that place the protein in the center of this box, and there should be at least minimum of 1 nanometer distance from the edge of the box. Okay, so from the edge of the box there should be 1 nanometer distance in this case. So you can change into 2 or 3, whatever you want, but do remember, the bigger the box size, the more amount of solvent molecule will be there, the more amount of time it will take to calculate all these things. Okay, so use the boys very precisely, or the amount required. You can you can do a trial and error. There are different kind of box, so that is defined by box type. You have cubic, you have tetragonal, you have octahedral. There are different kind of box available with us. So in this case, we'll just copy this command, second command, and we'll fire over here. Okay. So it has done all the calculations. So it says that this is the system size in nanometer, diameter, whatever the values are over here. In Gromex, whenever there is a pretty thought written over at the end of the output, it means that there is some kind of jargon statement made by any available person or any other event. But whenever you see this kind of message, it's the, like, it indicates that the program run has been completed. So whenever you want to see, there will be a kind of a thought written at the end of the output, and you will be written back to this command prompt. Okay, so now we have completed the second command, and now we'll move on to the third one, that is a gen box. Okay, so in this case, the third command is uh, Initially it was genbox, but now it has been replaced with a command called as solvate. This image is from the initial Gromax release, which was genbox, but now it has been replaced by a command called as solvent. So just copy this file. I'm just copying the third command. I'm just replacing over here. Okay. Okay. So the output of the second command is now we are using as an input of the third command. Ignore this right now. This is about water molecule, the configuration file for the water. And then this is the output, that is now where we will be having a protein under solvent. And then this is a topological file which we have generated initially. Okay. So I'll just open up Chimera and show you how all this looks like. The Chimera, and I'll just show you now how the whenever you're proceeding, how it looks like. So I'll just open the protein.grow. So this is the output of the second command. Okay. So this command out, sorry, this is the output of the first command. It is, I'll just open up over here. It's a protein file only, but it's a Gromax compatible file. Now I just wanted to open up the third file, that is protein box. So it is again a protein. You cannot see any kind of box, but if you open up a VMD, that is again a visualization, you will see a pretty box over here. And now I'm just turning up the third command and I will show you how it looks like. So I'm just copying this one. Okay, so now we have just run the third command and it says that there is a box has been created wherein we have added a solvent molecule. So the number of solvent molecule added over here are 30,291. A protein have 305 amino acid and this is the amount of water molecule. Obviously the water are very small molecule and there is a box. So this amount has been used to generate or to fill up the box. Okay. There is a algorithm and it's a randomized process. So second time if you do the same job, the number of the water molecules added would be pretty same, but the location would be different. But we don't need to worry about it right now. And this is the exact volume and how much densities has been occupied. And the output file has been generated. I will just open up the file and show you now how it looks like. So it will, I think it will take 2 to 3 seconds to open this file, perhaps more than that. Okay, till it open up, so I will just move on further. So now if you open up your topology file, now if you open up this topology file, and if you go, initially we had only one molecule, that is protein, but now we do have a water in this case. Sorry, we do have a solvent. In this case, it's a water. So we have this much amount of water molecule present in the box. I think it will take some time. We'll just move on to the fourth command. Now, as per this chart, what we have done till now is converted the PDB into Gromax compatible file. Then we have configured the box. We have added the solvent. Now we need to add ions to neutralize the system. Okay. So now we need to neutralize the system. Okay. So over here, if we go further, so now there are two commands over here. The first one is the gromp one, which is the Gromax preprocessor command. So whenever you run this gromp command, you need to supply the command with the parameter file, that is MDP parameter file. So that is available here. You can just click over here. It will open up a by default file. I'm not going to explain you all these steps into detail, but important one. So for this particular command, it is using a steep algorithm. So it's a steepest descent minimization approach. This is the information that when you need to stop the minimization, this is a different cutoff scheme, Verlet. So it's out of the scope for this present presentation or present lecture, wherein it's very, It's very difficult to explain all of this thing, but as a first time user, I suggest to you that you don't need to change any of this parameter file. Just use it by default, whatever it is provided over there. So I have pre-downloaded these files and they are right into this folder. So I'll just copy them and paste it outside. Or in what I can do is I can download again for you. So for downloading, there is an excellent command called as wget and then paste the link. The file will be downloaded. Okay. So now over here you have this ions.mdp. I think it's taking a lot of time. Okay. Moving further, now you need to run this command. So I'm just copying this command, again pasting over here. So just replacing with the output file of the previous command, the name of the output file of the previous command. Okay, rest I'm keeping it as it is. So whenever I run this preprocessor command, a TPR file is generated, a topological resistant file. So this is named by ion.tpr. So it is minus O over here. We are supplying in that with the topological file, which we do have generated from the first command, this one. And this would be the file which will be utilized for this command, that is protein.sol, which is generated by the third command. And this is a parameter file which you are feeding into this command. Okay, so just copy it and just run for you. Okay, so now it has run, it has prepared, or it has configured the initial file. It has pre-processed the initial file, we can say it, and then we'll run the next command, that is a generate ion command. Okay, so this you can paste it like this, so you can see it's a together command. You need to run it together, the first one, second, third, the fourth one, which includes two command. So now again over here we are using the same file, but now we will be producing an output file called as protein solvent with neutralizing ion, and then again topology file. And now, as I told you earlier, we can use NaCl. So we have plus, that is positive ion, Na, and Cl as a negative one. Either you can look into the charge, which was initially -4 in our case, so you can add +4, or if you don't know the charge, you can just simply put this word neutral. So it will automatically determine what kind of ion it has to add to neutralize the system. I'm just copying it and running it over here. So now there is a tricky, not tricky, but there's a prerequisite point over here. So you have created a box, you have inputted a protein, you have surrounded it by solvent molecule. Now, if you're going to add a chlorine or a Na into that system, before doing that, you need to remove the same amount of solvent molecule from the box. So for example, let's say this is a box. Okay, I have prepared it. Now I need to add some ion to neutralize the system. So for doing the same, we need to remove something from the box. So if we are going to add 4 ions, then we need to remove 4 water molecules from this box. So the system is balanced. Okay. So now this is asking to you that it will try to add 4 Na. The charge was -4, so it is trying to add 4 positive ions into your system. And it is asking that before adding to it into the system, what do you want to remove? So obviously we'll remove solvent. We are not going to remove the protein. We cannot disturb the protein. So in this case, we'll just select 13 and press enter. Okay. So it has replaced a solvent molecule. This is a number of molecule which has been replaced. It is a very random process. It doesn't actually specifically, it doesn't target anything, but it does in a way that this water molecule or these ions would be a little bit far from the protein. They're not going to interact with the protein. So it has replaced 4 water molecules with 4 sodium ion. So now you can see there are a lot of output file generated over it. So this is our final file for the step number 4. The first was protein, this one, then protein.grow, then protein box, wherein we have configured the box, then protein with the solvent, that is the solvent in the box along with the protein, and then you have ions to neutralize the system. I will just check it. Okay. I think again it will take a time, so perhaps we'll proceed further. So now you have, okay, it looks something like this. So there is a protein, this box, this blue one are water molecule, and these are the ions added to neutralize the system. So now you have a model system ready with you. Okay. So now the actual dynamic starts. So over here we will start with this first step, that is minimization. Okay. So again, there are two sets of command. One is a gromp, there is a gromex preprocessor, and then there is a mdrun. For this combination of for this gromp, we need to supply it with a molecular dynamics parameter file, that is energy minimization.mdp. I'm skipping this, but you can read later on. You can just remember this link, or I will give it to Kalyani. She will deliver it to you. So you can just go in, you can read line to line. I'm not taking time to explain this. I'm just downloading this file. Again, I'm opening up to show you. So this is a parameter file. So anything beyond semicolon is a comment which you don't need to worry about. The main thing is before this colon. So this is the actual parameter which we need to use it. Okay. You can modify this if you are expert into that, but as per my advice, just go with the by default parameter. So just I'm downloading this file. Okay, so now I have the MDP file. I'm just copying this for command. Okay, I'm replacing this with the name which I have used initially. The name, it's not necessary to use protein. You can use any other name. But why I'm doing it that because you can save the list of command and you can fire it very easily without going to that tutorial. So if you keep different names for each run, then you need to, every time you need to go and type in. So just use protein name by default. So you can use the same set of command for your every another simulation run. Again, similarly like this, which I explained to you, we are supplying an MDP file over here by -f. This is the file which we are going to use as an input. Again, a topological file coupled to that. And then again, we will have an output. Now remember, whenever we are going through this series of command, the topological file, that is topol.top, gets modified each time depending on the step. It will get a modification. So I just forgot to show you that how the topological file looks like after addition of ions. So just go at the bottom of the file. Initially we had only protein, then we added solvent, and now we have sodium ion into it. So it file says that there are 4 sodium ion. Properties or parameter for these are stored over here. Parameters for this are in over here, and parameter for this all are over here. Okay, so all of these are the important information. Now moving further, I think I'm okay. It looks something like this. Okay, so you can see this red, white, tiny molecule. They're water. They're in the box, and then this center over here, there is a protein. I am just doing presentation from my laptop, so it's not pretty powerful to showcase you this. But if you have a good computer with a graphics card, then you can easily visualize this particular file. Now moving further, what I've done is I will run this command. Okay, it has run, and now I will run this next command, that is an mdrun. So now over here, this is a gromex. It indicates that it is a gromex. Then we have a mdrun. Over here we are using -v, which is a verbose, so it will show you the output. And then there will be a lot of files generated over here, but we cannot define the name of file. If you don't want to do that, you can just use hyphen definition name. So all of the file will be generated by a prefix called as em. So em.tpr, em.trr, or whatever the file may be. Okay, I'm just copying this command and pasting over here, running it. So it will take a little bit of time. So there is energy minimization going on right now. And if you see over here, there will be a lot of files generated over here. So see em.edr, em.tpr, em.trr, and so on. So that's the reason why we have used -deff_nm. That is a definition name. That is em. So all of them will have a prefix of em. So right now we are over here this step. And similarly, we need to do the equilibration step, and then we can go for the production run. Okay. So just to simplify it, what I'm just going to tell you is this is a set of two commands which we have used for energy minimization. Then there are certain tools or certain program to just check it whether you have done it successfully or not. And then we proceeded to equilibration. Okay. So these are the major steps of any simulation. Right now we are over here, that is equilibration. So the first we'll go for NVT equilibration, that is constant number of particles, volume, and temperature. And then in the next step, we will go for NPT equilibration. So the parameter file over this NVT is over here. It's difficult to explain you all of this thing right now, but it says that the number of steps are 50,000. This is the integrator which is used in this case. Initially we used steepest. Now in this case we are using MD. This is a cutoff. These are the kind of, you know, Coulombic type calculation, that is particle mesh evolved for electrostatic energy computation. This is the temperature in Kelvin, which we are using it right now. So we'll download the file, same, we will run a gromp preprocessor command, and then we will run the mdrun command. Okay. So all of this command, I'll just copy it and show it to you. I just forgot. So over here it was EM. Okay. So all of these three commands are same in terms of syntax, only the files are different and the purpose is different. So over here, instead of using a minimization MDP, we are using NVT MDP, which we can download from the website. The output of this em.grow, we are using here as an input. Again, another restraint file. Then we have a topological file, and then this will be the output. And then again, run with or couple this command with mdrun, which will do a final run. Over here again, we are defining it by name NVT. The same set of commands now would be utilized for NPT equilibration. So again, the same set of command over here. So this is the same set of command. Okay. So instead of NVT, we have NPT over here. And after that, final simulation, again, the same kind of command. So again, gromp and again, mdrun. So you can see over here, if I just wanted to show you like this, so for this PDB to GMX, we have this one. For edit conf, we have this one. Then solvate, that is a replacement of genbox in the newer version. And therefore we have genion. So for genion, again, we need a gromp genion, that is actual command, that is generate ion. Then for energy minimization over here, we do have a gromp and mdrun. Then for nvt and npt, we do have these two commands, these two. And again, for the final run, we have this one. Okay. So these are the set of basic command utilized to execute the entire MD cycle using a GrowMax. If you use a different software, it may differ a little bit, but the flow chart or the flow remains same. That is, start from the coordinate file, generate a box, add water, minimize it, add neutralizing ion, then minimize it, then do a equilibration at a constant pressure, temperature, and number of particle and volumes, and then go for a final MD run. So this is all about it. Now, if you see this command, it has finished. There is a pretty thought over here. Water is just a water. Now, over here, you can also generate a graph and visualize it, whether there is a stabilization into it. So we'll just see the graph, how it looks like in energy minimization. So it started from here, the potential energy, and it decreased it, and then there is a constant plateau formation over here. So this over here, it has been stabilized over here. So if we are not extrapolating graph over here, but it would be something like this, a line over here, straight line over here. And this indicates the number of energy minimization step. So right now in this case, we have forced the program to not go beyond 1000. So that information you can actually see over here. Where is the file? File. So over here we have defined that the number of total energy minimization step should be no greater than 1000. So if we use 2000 or 5000, it will go up to the point where the energy has been stabilized. Okay. And this is the maximum number of steps. So it should not go beyond this. But if we talk about step, it is 50,000, and the step size is 0.01. Okay. And these are other parameters which we don't need to go into detail. So now if you do the equilibration, the same, we need to do it gromp, and again we need to do it mdrun, but it will take a pretty lot of time to do that. So can I skip this, Kalyani, generating this files?
Yes, please. Yes, please. We are
Yeah,
on a
okay.
strict time constraint. So could you please skip the part that's not really relevant?
Okay, so this is the same, it will take a same iterative part. And then when we jump to the analysis part, so it's a basic analysis they have described over here. Okay, and you can run this analysis and you can like what are the question you need to answer, depending on that you can do analysis. There are a lot of analysis command in Gromax manual, you can just read into it or simply just Google it, you will get a lot of commands. If you don't know anything, just refer to a paper, any paper, standard paper, MD simulation paper. You can see what sort of analysis they have done. And then there is an excellent Gromex forum. You can go into that. You will see what are the different function of commands, or what are, or how this command can be utilized for your purpose. Okay. So it's very difficult to explain all of the commands over here in this session, but what I what I suggest to you is that you just start using it, then only you will get to know it. Even I don't know all the sets of command available into Gromex right now. I only use a specific set which is required to answer my particular question. Okay. The first command is to convert a trajectory. So now after doing all of this thing, what we get is a trajectory. So trajectory file, a trajectory is a file which includes all the information about the coordinates, the energy, the motion, that is the dynamics into it. So this trajectory file is very important for us. So the first one is to process the trajectory file, correct for any periodicity, and if you want to alter it, you can also change time unit, the frequency of the frame, etc. So if I need to explain this command, the analysis command, again the analysis command are run by GMX, and then you have this trajectory conversion command, wherein this is the input of the this one last command, the input is the output of the last command. And then you need again provide with the file which is called as an XTC. If you really want to learn what are this file about, XTC, TPR, TRR, or trajectory file, or whatever the case may be, you can just go or Google it, you will get information about it, or you can just go into the Gromax software page. There is a description over there in the manual. You can do that. Okay. So this is the actual trajectory file which we have got after successful completion of an entire MD run. And then you need to use it. You need to correct it for any periodic boundary condition. Now, if I want to explain you the periodic boundary condition, it's a little bit tricky, but I will just try to explain you. Okay, so over here, we do have a protein, something like this. We have generated a box, okay? And there are solvent molecule over here. Now, whenever you give, you fire an MD, all of the atoms or molecule are in motion. How they will behave in that motion depends on the force field applied to it and the surrounding molecule, so surrounding atoms. So depending on that, it will behave. Sometime it goes out of this box, the protein may go out of this box, and it may jump out of this box. So what we really do is we use a hypothetical box scenario where you have original box which you have created over here, then you have a virtual boxes over here everywhere. Okay, we have boxes everywhere. So now what happens if this box goes out, it will go into another box. But technically, if you don't correct the periodic boundary condition, what you see is protein moving out from this on right hand side and again entering it from the left hand side. So we need to correct this. We need to remove all of this thing. We need to only center the molecule into one box, that is a center box. So it doesn't look to you like that the molecule is jumping out from one box to another one. Okay. So for that, this is a command where we are saying that please remove any kind of or correct for any kind of periodicity, and please center the molecule into that box. So if you fire this, you will get the output. I just see if I have a pre-run. I might be having somewhere. Just let me check. Karani, just wait a minute. Huh?
Sure.
So, sorry for the mistake. I just forgot to copy the file from my server in my lab. This is my own personal computer. Sorry for the same. So, the first command is for RMSD. It's a root mean square deviation. And this command is often employed to check the stability of the entire system or entire molecule, let's say in this case. So we can use this command. You can just read more about RMSD. You can just Google it. It's difficult to cover all this thing in detail in this a short time. So what it does is it takes the file for backbone. We generally calculate it. Why backbone? Because protein has amino acid have a side chain that is R, and R is very flexible in nature. So we don't include that R for this calculation of RMSD. We just go with the backbone. So the backbone is actually the peptide bond, the C alpha, the C, and the N. It doesn't include the side chain. Okay. So when we do that, we get some kind of RMSD graph, something like this. So I just wanted to show you a graph from my paper, one of my paper. So uh, actually this was not a correct one, but there was some problem over here. But you can see on the left hand side, there is a nanometer, RMSD in nanometer, that the unit is nanometer. On the right hand side, on the x-axis, there is a time scale. So now whenever we do an MD, we need to define a time scale, 100 nanosecond or 50 or 10 or 20 or 1000 or whatever it may be. There is no thumb rule for how much nanosecond simulation you need to run it. But in general, people say that run to a time till the RMSD gets stabilized. Okay. So in this case, let's say there are two RMSD. Like what I have done in this case is I have plotted RMSD of two different simulation. One of apoprotein and another one is for protein in complex with the ligand. Okay. So the black one is for apoprotein and the red one is protein in complex with the ligand. Over here you can see that it started from here around 0.125. It went. So these are all the fluctuations. Okay. And it went it and it got, I cannot say it got stabilized, but just to explain, if you extrapolate, it is a little bit stabilized over here. So this is a zone where it has been stabilized. So I need to get an average. I can calculate that. For plotting this graph, before plotting it, you need to run this kind of command. And you can obviously plot this graph using using a program called as XM Grace or Grace simply. So you can just install XM Grace and you can use that to see how the graph looks like. So whenever you run this command, you get an output file called as XVG. You just open up this file with any plotting tool. It will open up something like this. Okay. So whenever you open up, it's now in this case, we can say that the apoprotein is stabilized a little bit. So you can see there is a stabilization over here compared to the ligand, okay, compared to the protein ligand complex. So over here you can see there's some kind of problem over here. This is for another protein. So again, you can see this is for apoprotein. This is for RNA dependent RNA polymerase, and this one is for RNA dependent RNA polymerase in complex with remdesivir. Okay, so you can see this red one is pretty at a lower range, 0.5 fluctuation. So as a rule, do remember that the lesser the amount of fluctuation, the more the stable the system is. Okay, if there is a higher fluctuation, then the system is thought to be more unstable. Okay, so in this case, you can see over here the red one is a little bit stabilized compared to the black one. Okay, so there are a lot of different kind of graphs, or a lot of different kind of analyses you can use in to just see or to answer your question. One important one is RMSF. So that is root mean square fluctuation for each and every amino acid. You can record it. Okay, so let's say I have just used that RMSF graph over here. So this is the residue number. Okay, it is up to 305 in our case, MPRO. So for each residue, it tells you the fluctuation from its original position, or it not original position. It tells you the average fluctuation of your amino acid for, throughout the entire trajectory. That is throughout the entire 15 nanosecond simulation used in this case. Okay, so the black one is for apo and the red one is in complex with protein. Now, if you want to really see the difference, the pattern, overall pattern is same. You can see the same amount of fluctuation is present for this residue in case of apo and protein complex, but there is a difference over here. So this is black one is for apoprotein and the red one is for protein ligand complex. There's a difference over here, though the difference is very minute in this case, but then there is a difference over here. Now you need to really check out this residue individually into the PDB file. Is it a part of any, or is it a residue which is interacting with the ligand? or it is a residue part of it's a residue which is a part of active site or residue important for catalytic function of that particular protein. So this way you can make some kind of assumption. I'll just show you a assumption which we used. Probably I think it would be over here. Not um in this case I think you can check it out, this one. Okay. Okay. Now see, I have what I did in this case was I just simulated a SARS-CoV-1 monomer of Mpro and SARS-CoV-2 monomer of Mpro. So these are two different Mpro, though the protein is same, but it is from the original SARS-CoV. There is a first strain that was an outbreak in 2000, and then there is a second one which is outbreak in 2019. I was just trying to compare both of them. So you can see there is a large amount of difference over here. So there is a large amount of difference in the fluctuation over here. These are the important residue involved for its function, catalytic function, we can say. So you can see over here, there is not much difference, but there is a little bit difference over here. This is a great difference over here, but this is a zone where is a large difference over here, and this is a domain one. And this domain one is involved, up to the best of my memory, I think it is involved in dimerization of this Mpro. Mpro is generally found to be dimer in solution. So what I remember right now, it has three domain one, two, three, and this domain one is important for dimerization. So if you see the fluctuation over here, so this is the domain one, this is the domain two, and this is the domain three. So domain two, this one is the part where it interacts with the ligand. So this, the width of the tube indicates that how much fluctuation is there. So the more the amount of width, the higher the amount of fluctuation, the lesser the one, the thinner the one, the less amount of fluctuation over here. So this is also a kind of analysis which I used for my particular problem. So as I told you previously, there are a lot of softwares used, a lot of analysis program or script which can be used to address your problem. Okay. Okay. I think Kalani, I can hand over here. I think it's been overloaded for students, but I tried